Thanks, Stephen. I'm going to share my screen here, but while I do, I will thank you for inviting me to be a part of this seminar series. I've really enjoyed hearing the previous talks in the series, and I look forward to those coming up. So today, can everyone see that okay? And it fits in your screen and all the things it's supposed to be doing? Okay, great. So today I want to share some examples of specific ways we've used genomics in conservation management for endangered species in situ, ex situ, and somewhere in between. And I'm going to focus on the practice part of conservation and practice because we are never 100% sure, 100% confident in what we're doing. So we use the best data we can to try to come up with applications that will benefit the species. But certainly this is always a work in progress. So for the past half century and probably beyond, it's been recognized that the rate of species extinction is increasing and that there's a lot of species in imminent danger. In the most comprehensive assessment of its kind, the IPBES report on the dangerous and unprecedented decline of species worldwide and the acceleration of species extinction rates. So the report, which was published in May of 2020, paints a pretty ominous picture. And, we've all seen the headlines finding that around 1 million animal and plant species are now threatened with extinction, more than ever before in human history. And although it might sometimes be neglected in policy and practice, we all know that genetic diversity is an important component of biodiversity. Robust levels of genetic variation in a population reduces the negative effects of inbreeding. It provides the raw material for species to adapt to changing environmental conditions. It supports community and ecosystem functions. And it provides the foundation for plant and animal breeding to the benefit of agriculture, forestry, fisheries, and so on. And there have been several major advances that have allowed us to peer deeper into the evolutionary mechanisms that generate and maintain genetic variation, which has yielded, a ton of insight into biodiversity conservation and highlighted new opportunities for how we might do it better. So first has been the increasing use of creative sources of DNA, right? So if DNA can be found in nearly all cells, then we can extract it from samples of feathers, hair, or scat that can be collected without handling the animal at all. We can sample DNA from museum specimens, from forensic samples, or commodities sold at markets. In one of my favorite examples, researchers have undertaken mammal biodiversity surveys by utilizing DNA extracted from carrion flies and leeches, which is a truly innovative and creative approach. The second major innovation has been the advances in genomics and data science. We're now able to sequence far more of the genome from many more individuals than ever before, and to compare them across broader scales and with greater depth and greater insight than were previously possible. So we've also taken advantage of a rich foundation of evolutionary theory and detailed experimental work, often using model systems to explore the basis of conservation management approaches. So these laboratory experiments have been really useful, but often they can't provide sufficient insight into conservation of endangered species, many of which are vertebrates with small population size, a history of declining numbers, and often quite unique characteristics. So these attributes necessitate studying the species itself. I'm not sure how you could use Drosophila to learn a whole lot about this turtle species here, but I want to know more, right? So, but in turn, studies of these really unique species contribute back to our understanding of evolution, adaptation, and conservation management. Our lab is broadly based in molecular ecology, as Stephen said earlier, often with a goal of improving conservation and management. So we use genetic and genomic tools, modeling and bioinformatics in non-model species. We mostly work on mammals, although non-mammalian vertebrates occasionally make their way into the lab, and we've learned a lot from those experiences. So in evolutionary genetics, we sample, broadly across the landscape, across time, and across the genome to explore phylogeographic patterns and the mechanisms that generated them. We also apply genetic and genomic tools to wildlife management. One recent focus has been in the area of disease risk assessment and understanding transmission dynamics. So we've used genomic approaches to learn a little bit more about how plague is spread among prairie dog colonies and how that might be affected by vaccine bait distribution. We've also used genomic, we're currently working to develop a genome-wide suite of tools to quantify susceptibility of white-tailed deer to chronic wasting disease, which is a contagious neurological prion disease that's fatal to cervids. And in the context of conservation and captive breeding, our lab focuses on the conservation and management actions and their impact on populations, either realized or predicted. For example, we recently undertook a study where translocated desert tortoises survive really well in their new environment after they've been translocated, but the males aren't breeding and their genes are not being represented in the subsequent population, which has potentially catastrophic consequences for species recovery. We're also integrating genomic data into pedigrees to better manage captive populations held in zoos or semi-wild populations. And much of what I'll talk about today will be focused on this sort of right-hand side of things. So today I'm going to share some examples of how our lab has been using genomics to improve the conservation of endangered species so again, much of what I'm going to talk about today is ongoing, right? So I think that's helpful in illustrating that the hard work of conservation is, rarely ever finished, but, is a work in progress. So the first example I want to talk about today uses genomic data to improve captive population management. So in working with captive population managers, we realized that we could potentially make some significant improvements to the management of captive populations by incorporating genomic data. So we set out to develop a pipeline and an infrastructure to build that capacity. So there are a lot of ways that you could use genetic and genomic data to inform captive population management. Taxonomic identification is a very common application, including species or subspecies classification. So either assigning animals to known taxonomic groups or defining those groups themselves. And two, detecting hybridization. Because taxonomic delineations are not always clear and animal origins are not always recorded, this is really helpful for population biologists to make decisions about how to structure cooperative breeding efforts. Understanding heritability and hereditary disease is important for sustaining healthy and viable populations. So we might use genomic data to develop diagnostic tests to establish presence or absence within individuals, or population level studies of transmission dynamics. Disease heritability is certainly of a considerable interest to make a determination about whether or not individuals should be retained in the breeding pool, but it's not been widely applied yet, except in cases like California condor chondrodystrophy, where the disease is caused by a single mutation at a single gene, so it's pretty straightforward to monitor. So lastly, we can use genomic data to resolve incomplete or poorly known pedigrees. So maintaining long-term genetic diversity relies on breeding strategies that dynamically select pairs for breeding that exhibit, on average, low pairwise kinships with the rest of the living population. So that means they have few relatives in the population, or that their genome is underrepresented in the population. For example, breeding the green and orange elephants in the example. Incomplete or unknown pedigrees result, in an inability to accurately calculate kinships. And so it, hampers genetic management. And this is an absolute crisis in captive population management. Of nearly 500 species survival plan species, those are species which have a captive breeding program plan in place. 78% of those have incomplete pedigrees, and 25% have no pedigree information at all. So the reasons for these incomplete pedigrees include poor historical record keeping, right? We haven't always been able to keep good records, hundreds of years ago. uncertain parentage and group managed species, so species that are managed as herds or flocks or colonially, unknown ancestry from animals that are acquired from private sources, and ambiguity surrounding the assumption that all the founders are unrelated, right? Because we don't really know if those founders were unrelated or not. We can use analytical assumptions to fill those pedigree gaps, and we do, but they might be incorrect and are often based on worst case scenarios. For example, animals that came from a similar source, so a similar facility, or whether private or captive facility, might be assumed to be full sibs, to be extra cautious. And those individuals with unknown pedigrees might be excluded from breeding despite being potentially valuable to the managed population. So to illustrate this problem, I want to take a look at Nabalam, which is jaguar number 1003 down at the bottom. And by way of a quick reminder, in this pedigree, circles represent females and squares represent males. So the parents and grandparents of Nabalam are known, but ancestry farther back in her pedigree remains unknown. So jaguars 381 and 289 were captive-born animals that were acquired from undocumented sources by AZA facilities in 1972 and 1965. Jaguars 405 and 247 were born at AZA facilities in 1969 and 1962, but their parents weren't recorded. And the great-grandmother of Nabalam, who is 289, is particularly complicating because she had 15 cubs at the same AZA facility between 1968 and 1972, and paternity of those 15 cubs was not recorded. So we have no idea if they're full sibs from the same father or half sibs from different fathers. We also don't know if those fathers sired cubs at other AZA institutions, which would further complicate those relationships. So in short, because of all of this uncertainty introduced into Nabalam's pedigree, we cannot with any confidence identify Nabalam's living relatives, and thus Nabalam is excluded from breeding because population biologists can't identify a genetically appropriate mate for her. So if we could use molecular data to resolve those relationships, we could reincorporate potentially valuable individuals back into the breeding population. So one way to do that would be to resolve unknown parentage. So in order to do that, though, you need to sample the offspring of interest and all putative parents. So there's a lot of sampling involved. Also, the analysis might not be effective. For example, if you have unknown parentage deep in historic pedigrees and potential parents are deceased. And you need to resolve parentage every generation if that problem is systemic, right? You would need to continue to do this every generation. So the other way you could do it is you could empirically estimate kinship. To do this, you need to establish a pretty comprehensive baseline of the relationships from which all living animals descend. So that requires fairly substantial sampling across the entire ex situ population. But then these molecularly derived kinships can then be used in lieu of the pedigree-based kinships for breeding pair selection. And historically, we relied on small numbers of markers like microsatellites that produce pretty inaccurate estimates with high sampling variances. So today, the fact that we can generate large panels of SNPs for non-model species that produce very accurate estimates of kinship really makes this a viable option. So we set out to develop an easily accessible workflow for integrating genomic DNA into breeding pair selection so that we can improve pedigree-based management. So we developed our approach and we tested it in 5 species, 2 of which I'll show you today, and then created and provided all the necessary infrastructure, so protocols, software, training, et cetera. And then each of these SSPs we chose to validate our approach because they had their pedigrees unknownness distributed differently. So in koalas, although the contemporary pedigree is very well known, the kinships among the original founders are less well known. So only 39% of the living population can be traced back to the original imports. And not a single living animal can be conclusively traced to a wild-caught animal that's known to be unrelated. The Fennec Fox is a popular animal in the US pet trade and can be legally owned throughout much of the country. So as a result, many of the foxes in the currently living SSP population are of unknown origin because they were surrendered or confiscated from private owners. And because there are a limited number of Fennec Fox breeders in the US, we believe that many of the SSP animals that originated in the private sector may be related. So an early obstacle that we encountered with this project was sampling. Animals are quite difficult to sample, not only because every zoo has different procedures and animals can only be sampled oftentimes during their annual exams, but also because some animals, including those original founders, have been dead for many years. So we recognize that one of the community's needs would be a way to prioritize sampling in a way that ensures that we sample the most critical animals, but don't waste resources sampling genomes that we've already sampled, for example, parents and their offspring. So we built a software that prioritizes sampling in order to maximize the completeness of the pedigree. So it ranks individuals by the proportion of the pedigree that the animal would help resolve, and then iteratively re-ranks as you sample so that you don't duplicate effort. So it's a really simple program and not fancy by any stretch of the imagination, except for the Tern, of course, who is very fancy, but is really useful to make sure that you're sampling thoroughly and also not redoubling your efforts. So now that we have the animal sampled onto the genomic data, so we used DD-RAD, which is a modification of the RAD-seq technique. And we chose this approach because for a couple of reasons. One, because we often don't have reference genomes, although in these two cases we happen to have them. But also because it uses 2 restriction enzymes to fragment the DNA. And so the idea, one of the reasons to do that is to increase the chances that you'll sequence the same loci in multiple runs in case people needed to run samples at multiple time points. So we used both reference-based assemblies and de novo assemblies. The reference for koalas is pretty good, but it's, let's say, less than ideal for Fennec Fox. So as you can see, there's more missing data with de novo compared to the reference-based assembly, but there are more individuals retained and more loci. So today I'll show you the results from the reference-based assemblies. So what we did first, and what I'll show you today, is correlate molecular kinships with pedigree-based kinships. And so if you look at these data, what you're hoping to see is a really strong correlation between the molecular-based kinships and the pedigree-based kinships, because in koala, I said we know a lot of the contemporary pedigree. It's pretty well known. And so here you can see that they correlate pretty well. The pedigree-based kinships correlate well with the molecular kinships. You can see the same data on the right in distribution form. You can even start to see relationship categories. So you can see like there's a bunch of dots right at 0.25, at 0.125, et cetera, which is really promising. And, again, this is in large part because most of the unknownness in the koala pedigree is concentrated further back in the pedigree. So we have pretty good records for the contemporary population. So these results suggest that the data being used to manage koala breeding is pretty accurate. But we can use some of this molecular data to help refine our estimates of kinship. When we look at Fennec foxes, it's a little less clear. So the proportion of the pedigree known in this population is only about 10%, so there's a lot more unknown relationships. But you can see right away that the correlation is pretty low, driven at least in part by a large number of individuals across the bottom axis. So these are individuals that are assumed to be unrelated, but based on molecular data are related to varying degrees. That's reflected in the distribution data as well, where there's an excess of 0 values in the pedigree data in blue compared to the molecular data. So we have a framework for comparing molecular and pedigree-based kinships, and we're working on getting those integrated into the pedigrees to maximize the collective information we have from different sources. So we've done this PedSam software to help identify samples for collection. We've sequenced the samples, correlated those with kinship values, and now we're working kind of on this last stage. And one thing that's been really important to us has been to be able to make this as an easily accessible workflow and pipeline for zoo biologists. And so we have software and we have guides to carry them through this whole process so that they can undertake this for their species of interest. And so we've given a bunch of training programs and things like that as well to help people who are actually making the decisions about captive population management so that they can do it better. So in the second example of genomics for improved conservation, I want to share some work we've done in bison. Bison are an iconic species in North America and heavily managed. But even in such a classic system, there's room for improvement. So we set out to use some of what we had learned in captive populations and apply it to these intensively managed, we like to call them sorta situ populations. So as habitats are increasingly altered and wildlife populations are more heavily impacted by our activities, the intensity of wildlife management has increased. So there's more and more species that are dependent on regular monitoring and intervention to ensure their persistence. So practices like moratoria, anti-poaching efforts, predator removal, culling, so removal of animals, healthcare, disease management, they're all often undertaken at the scale of the individual animal. And those intensive management activities are often, go hand in hand with a decrease in space where populations are becoming smaller and more isolated. And, as we all know, small and isolated populations are not only less demographically stable than large populations, but more susceptible to erosion of genetic variation by drift, which goes unmitigated by gene flow if they're isolated. And so as a result, preserving genetic variation has become a priority for management, particularly for these small and isolated populations. So there's a number of different strategies to maintain genetic variation in small and isolated populations. There are indirect methods that don't require genetic data that maximize the exchange of genetic variation from generation to generation. So things like maintaining balanced sex ratios, avoiding massive fluctuations in population size over time, and extending mean generation length are all things that we could do. For example, removal or contraception of young animals results in a greater proportion of offspring born to older females, an increase in generation time, and higher retention of genetic variation over the long term. So these indirect approaches are typically straightforward to implement because they don't require genetic data to be obtained for any individuals. But management strategies could also be designed to manage genetic variation directly. So if genetic data are available, strategies designed to maximize retention of alleles could maintain variation. So if rare alleles are found in individuals with underrepresented ancestry, an allele retention strategy could preserve rare and potentially important variation in a population. But as many of you are already thinking, if rare variants are rare for a reason, if they're deleterious in some way, selective retention of those alleles could reduce the population's fitness. And strategies that are designed to retain specific rare alleles might result in loss of genetic variation across the remainder of the genome. So in captive population management, the most effective management strategies have been those that consider genome-wide variation, specifically by minimizing mean kinship. So again, breeding individuals with few relatives in the population. But for wild or semi-wild populations where you can't dictate breeding pairs, you could remove individuals with high mean kinships. One example of that is in the endangered takahe in New Zealand, where they remove individuals with high mean kinships and replace them with unrelated individuals from a captive breeding program. That's a really interesting concept and has been able to retain genetic variation over a long term. That concept has also been evaluated as a possible option for controlling population size of wild horses on Assateague Island while still maintaining genetic variation. It requires the collection of genetic data, but if that data were available, it could be a really valuable strategy for conserving variation in intensively managed populations. So I'll talk a little bit about plains bison, give you a little background. Just 200 years ago, plains bison numbered 30 to 50 million animals in herds of up to 10,000 animals. But by the late 1800s, massive overhunting and land use change reduced the population by over 99% to fewer than 1,000 animals. So efforts to establish managed herds led to an increase in numbers. Today, there are about 500,000 bison, but most of those are in private herds under unknown management schemes. Only about 3%, or about 15,000 animals, are currently maintained in conservation herds managed by federal agencies and NGOs, non-governmental organizations. So the challenges are pretty well known, right? Herds were founded with very few animals. There's little, if any, gene flow among herds, in part because of, valid disease transmission concerns. And the herds are maintained at relatively small size through annual culling, because these herds exist on small isolated parks and reserves that have to accommodate multiple use goals. So we modeled genetic variation and inbreeding under 3 culling strategies. The first is a random culling strategy. So this is the indirect approach, right? Where culling decisions were based on maintaining balanced sex ratios and culling yearling animals to extend mean generation time. The other 2 strategies extend this by incorporating genetic information, but also have the balanced sex ratio and the extended generation time. So the second is a mean allele frequency, or MAF strategy. And this strategy is similar to what the U.S. Fish and Wildlife Service was using to guide their culling decisions. And it's based on that idea of maximizing allele retention, where yearlings with common alleles at a target set of loci were prioritized for cull. The mean kinship, or MK strategy, in this strategy, yearlings were chosen for cull based on their average kinship values, and that essentially equalizes founder genome representations by preferentially removing individuals whose genomes are overrepresented in the population as a whole. So we parameterized this model using bison biology from the 2004 to 2010 Fort Niobrara bison herd, which is in Nebraska. We used a target population size. In this initial model, this population happens to have a target size of 350 animals, to give you an idea of what population size we're talking about. We included sex and age-specific fecundity and mortality and a maximum age. You can see the mortality functions there on the right. We included the age range, a bunch of breeding parameters, right? So, one of the things that's unique, not unique, but one of the things to consider for bison is that male dominance exists. So we incorporated male dominance into the model as well. You can see that males peak in breeding around, 8 to 15 years or so. And so we included that too, some male dominance parameters, including the proportion of males that were dominant, the age range, how many years they were dominant in a row, and the proportion of offspring produced by dominant males. And we had genotypes at 55 microsatellite loci, so we included that, but certainly this model would work for SNPs as well. And nearly complete pedigree information, which is, handy to have. So the overall model flow, we chose breeders, produced offspring, calculated kinships at that time, completed culling based on whichever scenario, and then induced mortality and aging. We did this for 1,000 iterations of each modeling strategy, reported summary stats at 100, 200, and 500 years. And those summary stats are allelic richness, heterozygosity, gene diversity, and inbreeding for 55 target loci. And also we generated 55 non-target loci. So these were generated in the model just by taking the allele frequency distributions, by sampling those allele frequency distributions to create 55 non-target loci that were tracked and measured, but were not used to guide culling decisions, because we wanted to assess the impact of culling decisions on the rest of the genome. So here are the results of the modeling. So at the top, you'll see 3 columns of graphs. Each column corresponds to the 3 culling strategies. So we have random, the mean allele frequency, MAF, and MK. Data are provided for each of the 4 genetic diversity metrics. So we have allelic richness, heterozygosity, gene diversity, and inbreeding at the bottom. The dashed lines represent the target loci, and solid lines are the non-target loci. And as you know, GD, so gene diversity and inbreeding, are genome-wide measures, so they don't have separate lines. So the first thing to note is that genetic diversity decreases and inbreeding increases in all the strategies. And this is not surprising, right? This is expected because this is a population of 350, right? It's a finite size population. So after 500 years, the random strategy performed the worst, with the lowest values of allelic richness, heterozygosity, gene diversity, and the highest inbreeding. MAF retained the most diversity, but only at the target loci, because that's what it's designed to do, right? It's designed to retain alleles at target loci. So of course, heterozygosity is high, and of course, allelic richness is retained. But at the non-target loci, you can see it performs vaguely similarly to the random, only very slightly better than the random strategy. The MK strategy retained the highest diversity at non-target loci and had the slowest accumulation of inbreeding. And also, although the error bars are a little hard to see, the MK strategy had the lowest coefficient of variation among the 1,000 model iterations. And so it was the most consistent strategy across model iterations. So it was the one that produced the most consistent results, which is great when you're trying to manage populations. So the results are summarized in the boxes above, but essentially show that direct management strategies that incorporate genetic data are better than those that rely on indirect approaches, though the latter, of course, are less expensive programs to maintain. And if you have genetic data, pedigree-based strategies are far better than strategies aimed at preserving variance at a panel of loci. And this is in line with captive population management strategies where breeding pairs are selected to minimize mean kinship. We've also extended this work to show that the MK strategy is consistently effective across bison herds with different founding sizes and current levels of genetic variation. So in order to help bison herd managers implement this strategy, right, because you have to actually give them, capability of actually implementing it, we built an application for them to use that allows them to enter their own herd parameters and to test out different management strategies. So among other things, it outputs a prioritized list of animals to cull. And again, it's nothing fancy, but the U.S. Fish and Wildlife Service is now using an MK strategy to manage their bison herds, which is really exciting. The software is quite flexible because it allows you to input a pretty wide variety of parameters without needing to rewrite any of the code, although you could certainly do that too. So I think it could also be adapted to a wide range of species. So the last example I want to talk about today is about some of our ongoing work in Hawaiian monk seal, a critically endangered marine mammal. So way back in the beginning of this talk, way back a half hour ago, we talked about the importance of genetic variation for biodiversity, for inbreeding avoidance, adaptive capacity, community and ecosystem functions, et cetera. So as species dwindle in numbers, populations become small and isolated, demographically unstable, susceptible to genetic diversity erosion. And a lack of, the persistent lack of genetic variation also creates two related problems for genomic data analysis. First, generating more sequence data or sampling more individuals may not actually reveal any more variation. You may have seen all of it. So when the number of variants is limited, evolutionary and demographic processes leave signatures on the genome that are undetectable, regardless of the resolving power of your genomic data set. So statistical tests that rely on polymorphisms like parentage, tests for selection, GWAS, aren't possible if polymorphisms aren't there. So for example, heterozygosity above about 0.2 seems necessary to resolve parentage, which is a value that's completely out of reach for a number of endangered species. Second, site frequency spectra will be skewed toward low frequency alleles that are more difficult to distinguish from sequencing error, which I'm sure many of you have encountered. Low frequency variants are likely to be removed during filtering for minor allele frequencies, which impacts downstream analyses, in particular, those that are sensitive to low frequency alleles. So things like, population structure, selection, or purifying selection detection, or population expansion. Filtering by allele count rather than by frequency, explicitly incorporating quality scores into MAF cutoffs, those will help distinguish errors from true rare alleles, but real biological limitations endure for low diversity species. Speaking of low diversity species, the Hawaiian monk seal is one of the world's most critically endangered marine mammals. The total population is about 1,400 individuals that span the entire Hawaiian archipelago. Subpopulations at different sites are impacted by localized threats and exhibit variation in demographic rates. Currently, most individuals, about 1,100, are distributed throughout the small islands and atolls in the Northwest Hawaiian Islands, and about 300 seals inhabit the larger and more populated or human-populated main Hawaiian Islands. In the Northwest, the seal population declined steadily from the species first monitoring way back in the 1950s through the early 2000s. Meanwhile, monk seals remained rare in the main Hawaiian Islands until about the mid-90s, and then rebounded pretty substantially starting in the early 2000s, for seals anyway. These patterns have led to an overall stabilizing trend across the species range, though local demographics and age-specific survival rates continue to vary between the regions. So comprehensive conservation efforts have certainly played a huge role in population stabilization, including protection of marine areas, translocations of aggressive seals, aggressive males, rescue interventions in case of injury and entanglement, disease threat mitigation, rehabilitation of malnourished individuals, ongoing monitoring, the list goes on and on. Yet questions remain, including some pretty fundamental to conservation that have been intractable using previously available approaches. These include things like basic levels of genetic diversity, population structure, migration patterns, the designation of appropriate conservation units, but also questions about why the MHI, the main Hawaiian Islands population, is rebounding. Is there increased reproduction within the main Hawaiian Islands, which would be great, or is there migration from the Northwest, or both? So we sampled 200 Hawaiian monk seals from across the archipelago. Our sampling was more intense in the main Hawaiian Islands because that's where the populations had experienced the population growth, and we were hoping to gain insight into the productivity migration hypotheses. We collected best rad data, which is a different modification of RADseq, and aligned them to the Hawaiian monk seal reference genome. Filtering was critical in this data set and also challenging, right? First, many samples were collected nearly 20 years ago, and several were of low quality. Second, filtering parameters were challenging to set because of the low diversity in the populations. So we were left with data from about 125 seals after filtering, and those are the sample sizes by island that you see on the map. We were able to identify a modest number of SNPs in our data set, but what they revealed is that genetic diversity in the species is outrageously low, among the lowest ever recorded for a wild mammal. So in our SNP data set, the expected heterozygosity was 0.034. That's right, 3%. So this value is in line with genome-wide heterozygosity based on the reference genome, which is the H value, which is 0.0001. So for comparison, that's a lot of zeros. So for comparison, that's about 10% of the heterozygosity seen in the reference genome of a Northern European human, and is identical to the heterozygosity observed in the critically endangered vaquita, which there are fewer than 20 left in the world, and which until now was the mammal species with the lowest recorded genome-wide heterozygosity. So despite all of this low diversity, our genome-wide SNP data set contains sufficient statistical power to delineate island-level structure for the first time! Exclamation point, this is great. On the left, you see a discriminant function analysis showing the differentiation between regions, Northwest Hawaiian Islands in blue and main Hawaiian Islands in red. You can see there are a few individuals from the main Hawaiian Islands that group with the Northwest, which could indicate migration or weak structure. Levels of genetic variation were similar between the regions, which suggests that although these populations are distinct, it seems unlikely that they've been completely isolated over the long term, because that isolation would be expected to erode diversity in the isolated population. On the right, you can see that our data set was even powerful enough to differentiate seals from different neighboring islands within the main Hawaiian Islands, so Oahu, Molokai, and Kauai. This is really exciting, in particular as we ask questions about the potential for inter-island movement within the main Hawaiian Islands. So the genomic data that we collected for Hawaiian monk seals have provided a lot of data critical for conservation, including how to define appropriate conservation units, some insight into regional population dynamics, some target islands to prioritize for translocations, and the foundational data to design a captive breeding program should one ultimately be necessary, or to design how to found that population, to maximize representation. But real limitations remain, right? So for example, understanding the variance in male reproductive success would help us better understand mating and determine whether the main Hawaiian Islands is experiencing population growth because of increased productivity, but all that mating occurs in the ocean. So we were hoping to be able to assign parentage to the pups that are on the beaches with their moms, but we were completely unable to assign parentage in this system, even to known mom-pup pairs, whether we used individual SNPs or microhaplotypes or any which way we manipulated the data. So there are challenges that remain even when using really powerful genomic tools. And, I think the overall theme of this whole thing, right, is, and this speaks to it well, is that that's where our creativity can really shine, right? Where we can integrate genomic data with pedigree data, or with modeling, or with ecological data, or all of the above, to really better understand the evolution and maintenance of biodiversity and how we can conserve biodiversity in a real way over the long term. So with that, I appreciate the support provided by these organizations for our project and others that target species conservation using genomics, and the members of my lab. And this picture always reminds me that I need a new updated picture of my lab, many of whom are here on the video today, who have tackled all kinds and continue to tackle all kinds of unique challenges in their work to promote long-term persistence of biodiversity. And so finally, I'd like to thank the EVOLTREE group, in particular, Stephen, Christian, Lidvina, and Santiago, for organizing this incredible seminar series. And with that, I would be happy to continue discussion if anyone has any questions or comments.
Great, thank you, Emily. That was superb. We give you all a silent round of applause. Thanks very much. Fantastic. Well, can I just, well, thanks very much again for the talk. I just want to highlight one comment that was made in the chat as you were speaking from Andrew DeWoody, just highlighting a book on the American Plains bison, which might be of interest if anybody wants to follow up on that story. And then I will now open the floor. So if anybody has a question, would you please raise a hand and we will bring you forward. Would anybody like to kick off? There's one here. Felix, please.
Yes, I can try and break the silence,
yeah,
at least acoustically. Thanks, Emily, from my side. Very interesting insights here. I wondered, in particular in the bison story, where you showed the effects of the different strategies for culling on, I presume, anonymous or even mostly neutral genetic diversity. So how do you consider this in terms of the adaptive genetic diversity, which might, or very realistically, behaves quite differently, and how this culling strategy might actually pick out individuals that in the long run would be beneficial for the population?
Yeah, I think that's the next step, right, is to actually consider the fact that these populations are adapting to their landscape and they're adapting to their environment, right? Not just the environment, or not just the landscape, but also the fact that they're contained. They're this sort of intensively managed, fenced in populations, but they're wild bison, according to our federal designations. So we're in this sort of no man's land of, they're definitely adapting to their new environment, but we're trying to preserve genome-wide variation. So right now, that step is a big one. And so I think that's where we are now. So meeting managers where they are is really important in this case. So I think, genome-wide variation preservation is a great goal, but I do think that, I'd be interested to see and know more about how these strategies impact adaptive variation. And so we could, very easily model that, but I'd also like to look at it in real herds. We did look at introgression. So there is some cattle gene introgression in some of these herds, and that's a concern for herd managers. They want, purely wild bison. And so we also tried to add into the mean kinship strategy like a secondary selection, not natural selection, but a secondary decision-making criteria that was based on their level of introgression. And it turned out that because we were really only dealing with the animals that are right around the cutoff for getting culled or not culled, it actually didn't affect genetic variation that much at all. But that was a pretty small thing to factor in. So I'd like to see it factored in sort of on a more fundamental level so that it was incorporated throughout.
So would it be an option to get started on for example, things like MHC diversity, that this could be a bit of a more directed criterion for such a selection or decision making?
Yeah, I think that would be a great first step to introduce into the modeling first and to show how it works or how it is impacted, and then to sort of build from that. I think that's a great suggestion.
Do you get any sort of trait-based assessment of these populations at the same time as you're collecting molecular data?
Somewhat. Sorry.
Trait-based assessment.
Yeah. No, there's not. So other than, they round them up every year and, mark the new ones and send them back out. Other than that, they don't really take a whole lot of data. The genetic data was only collected to get their pedigrees. So they were more concerned with a dominant bison being dominant for 10 years and siring all the offspring, which is definitely a concern. So the original genetic data was for that purpose. And so we saw an opportunity to be able to build the pedigree and then actually use it to guide culling strategies, thinking, ranking a list of animals and choosing the ones with the highest mean or with the lowest mean kinship for breeding is one way to do it, but you could also rank them and lop off the bottom if you had to, right? Which, I wish we didn't have to, but that's where we're at. Yeah.
Andrew, please, you have a hand up.
Yeah, hi Emily, great talk.
Hi.
Just one comment, one question, a comment. So I had a former PhD student, Janet Willoughby, who also did some work on SNPs, in the context of mean kinship and pedigrees and maintaining genetic diversity. And one thing that she did that you might consider in some of your work is to go back and look at the RADseq data and map those onto functional genes and then apportion them out, based on the likelihood of the SNPs being functional or not. But my question is really sort of a philosophical one about the Hawaiian monk seal data, because, I don't really know much about that system, but I do know enough biology to know that, the archipelago was probably only, an initial population that has since diverged over time, right, to these different islands. And so, yeah, you've got a lot of discrimination now, with your, with the SNP data sets. But, I'm always a little bit, in terms of conservation, I want to hear your thoughts on how important is that, right? If I think I heard you say there were like 1,600 monk seals, something like that. Does that discrimination really matter? And, with my own students, I always make the point that, in humans, we could, if I had a cancer, we could take DNA from the tumor cells and differentiate those from, my epidermal cells or my liver cells or anything else, just because of the somatic mutations that occur over the, a 50-year lifetime. So just because we can discriminate them doesn't necessarily mean that it's important in a conservation context. So, and I don't mean to pick on you, I'm just genuinely curious about your perspective on that, and how should that, influence Hawaiian monk seal conservation moving forward? Does it help or hurt to be able to, have that fine scale differentiation that you detected?
Yeah, I mean, that's a question for the ages, right? I mean, I think every conservation story, whether in plants or animals, has to ask that question. So I'm glad you brought it up. For the Hawaiian monk seals, each island has very different threats. So the main Hawaiian Islands, for example, things like toxoplasmosis is a huge problem. Feral cats have toxoplasmosis. They give it to Hawaiian monk seals, and they're extremely susceptible to toxoplasmosis. Also, with the increased human population, there are increased, let's say, direct human killing of monk seals that occurs in the main Hawaiian Islands that is not occurring as much in the Northwest. In the Northwestern Hawaiian Islands, there are populations like on French frigate shoals. The sharks there have less prey base than they used to, and they have become increasingly specialized on actually beaching themselves to get at the monk seal pups. And so it would be good to know, this resolution in this case would be good to know whether French frigate shoals seals are moving away from bad habitat to the main Hawaiian Islands, or if, for example, beach preservation in the main Hawaiian Islands is causing, increased productivity. So those kinds of questions are really important for Hawaiian monk seal conservation, especially because there is such an intensive conservation program. They are doing a lot. They go out on boat tours across the archipelago to monitor, to tag seals, to rehabilitate. They do a lot of this work. And so to figure out where to target that work, and to figure out if, are animals fleeing French frigate shoals? Are there islands where there's increased productivity? These would be really great things to know from an ecological perspective. So I think in this case, the genomic data is going to provide resolution that helps us target where to implement which conservation actions. And certainly, this is definitely a question that you have to ask in every case, because, in some cases it's going to have direct implications like that. And in other cases, you're right, it's, dancing around, what do they say? Like angels dancing on a head of a pin or something. So, it's kind of trying to, rearrange the deck chairs, right? And that's a really important question to ask as you go down this path, to collect more and more data to do what? So I appreciate that question. And certainly, back to your first comment, definitely Jana's work has come up in our lab meetings a lot, and certainly comes up in our readings a lot. That was really important work and really relevant to the stuff that we do.
That's a great answer on the monk seals, right, about all the different conservation aspects. So yeah, thanks for that.
Great, thank you very much. Is anyone else have a hand up? If not, could I just ask, sort of comparing this to the plant situation, when we've got, I guess, typically we're dealing with very much larger population sizes, and inbreeding's not so much of an issue, but there are a few instances of very small population sizes where there are very evidently hybrids which contain variation which come from now extinct populations. I mean, is hybridization talked about as an issue for these, particularly the very small population sizes that you have in some of these species?
Sure. Yeah. So, I think one of the best examples is probably, not mine, but the Galapagos tortoise example is a really good one, in that some of the remnant lineages are found in hybrid individuals. And so trying to determine what to do with the last remaining, genetic lineages that are introgressed in with something else, is a really important topic. I think, with, it sort of depends on whether they're wild or captive populations. I think that's, a distinction that's made, whether it's valid or not. I think if hybridization occurs in a wild population, I think, there's questions about, how it arose or where it's headed, or, maybe what ecological factors are at play. If it occurs in a captive population, there's a little bit more potential control over where to go with it from here on, right? You could breed it out or you could try to, make it more pure, or you could try to see, if there are fitness differences between the two. So, I think there's a little bit more experimental type work that you could do, or you could base some of your guidance on experimental work. Whereas in the wild populations, dictating that is a little bit more challenging.
No, absolutely. We just have some instances where there's a great deal of sensitivity about the idea of releasing something that was bred, even to rescue its genetic variation, but to release that back into the wild is a very sensitive topic. I imagine it's even more so for animals.
Yeah, absolutely.
Christian, did you have a hand up?
Yeah, that's just a very small question. You showed this graph where you compare genome-wide diversity among species, and I was just asking myself how much sense it makes to compare species genome diversity, given that we use different methods, we use different reference genomes, and probably different number of samples how to derive that. So I think it's really interesting, but isn't it? Is this also used to kind of I don't know, rank species priority programs or something? Because then I think that would be quite dangerous.
Yeah, no, not used, other than just to kind of demonstrate it's very, very low. But yeah, I think using these reference genomes, you're right, they're all generated using different numbers of individuals and different metrics, but they're looking at heterozygosity levels within a genome, and then averaging that, as you said, across individuals. So they certainly will vary, but it gives you an overall sense of where species lie in terms of where you might expect overall diversity to be. But you're right, if they sampled one individual from a population that's really isolated, then it's going to be lower than if they sampled from a more continuous population somewhere else in its range.
I've also seen studies that use these values then as a predictor for something else. And then I think it's getting a bit critical if you want to say something then.
Yeah, I agree with that.
Thank you.
Thanks, Christian. Anyone else? If not, I might ask one final question, if you don't mind, just about again, sort of making the comparison with, I guess, our forest world, where we're typically trying to manage, I guess, moving more and more towards managing processes, so that we're recognizing the fact that populations are going to be changing over time, particularly as the environment changes. So I just wonder how you see that balance between trying to manage genetic diversity and allowing populations to change in a way that allows them to track changing conditions. I guess it's a particular challenge for some of these populations, but well, how do you feel it's best done for critically endangered animals?
Yeah. My short answer is I don't know, right? I think we're all in that boat, right? I think when we get to the really endangered species or the captive species, the idea is to give them enough genome-wide diversity that they have a fighting chance, right? That they have some chance of making it to a place where they have standing genetic variation that they could adapt to some other environmental condition. So it's just trying to get them there. And it's less even a consideration about whether they will be able to adapt to a specific environmental condition, right? We don't know what that's going to be. I think, in wild populations, we're thinking about that a little bit more. But even still, if it's a critically endangered population, I think, we're more able to think about things like hybridization, right? Where we might use rescue to try to introduce more genome-wide diversity into the population to, avoid, inbreeding depression. But, the same argument goes toward adaptation, right? To give them variants that they might then be able to use. So I think that's sort of where I see the discussion happening in animals, but we're very much still in the midst of that discussion, which I think is part of, what binds us, right? The animal people and the tree people, we're all talking about the same things is, what about rescue? What about adaptation? What do we do going forward, especially, not especially, but in particular for some of these critically endangered species? Are we just trying to get to tomorrow? Sometimes, or are we trying to think about long-term persistence? And, in our lab, we're definitely trying to think about long-term persistence, but we don't always have the tools to be able to do it. And more importantly, we don't always, I suppose we don't always have the raw materials, right? We have very little variation to start with. So we preserve what's left and go from there.
Yeah, great. As you say, it's very much unresolved debate, I guess, even in our world. So.
Yeah, it was a very apt title when you called it genomics or genomics in practice, right? We're all practicing, which, is important, I think, to recognize.
Absolutely. There was a couple of comments just in the chat. One from Andrew. I don't know if you want to speak to this one, Andrew. Just to add a little more to, yeah, go ahead.
Yeah, not really. I think Christian's spot on, right? It is complicated to compare genome-wide diversity across studies, but I think there are a couple of groups that are sort of working on approaches to, to try and standardize something. So, yeah, I think it's an important question, but it's complicated.
Mm-hmm.
Yes, absolutely. Thank you. And Bruno, I don't know if you want to just raise the point you've made.
Yeah, hi everyone.
Hi, Bruno.
Yeah, just to say that in medicine, they also have the same problem of standardizing results across multiple tests. And so one way of doing this is meta-analysis, where you consider all the possible differences and you account for them in your analysis. So that could be one way also of comparing species genomes.
Bruno, your sound is a little quiet.
Sorry.
Okay, no problem.
Yeah, no, that's definitely true, though. I think that, trying to use a meta-analytical approach is a really good way to try to take some of those differences into consideration when you're trying to develop these species to species comparisons.
Yeah.
Thank you. Great. Okay, well, I'll have one final call. If there's anybody else who has another follow-up question they'd like to raise just now, please raise your hand now. If not, I think it just remains for me to say thanks again, Emily, for a really, really interesting talk. Really some critical issues that concern us all there, but yeah, really nicely presented, and thanks again for taking the time to speak to us.
Of course. Thanks so much again for having me and for putting on this whole webinar series, the series of webinar series. It's a lot of work and it's been really great to see all the talks, and I look forward to the ones coming up. So thank you for that.
Great, thank you. And that's a nice lead into just a final note to say we have one more talk in this series. Are you going to show a slide, Christian? It'll be from Louis Bernatchez. So the final talk is going to be on genomics applied to fisheries management and conservation this time. And I just want to highlight the fact that this one is next week, but not on this day. So it's on the Thursday. It will be on the 25th of November at the same time, but Thursday instead of Wednesday. So I hope to see as many of you there as possible. Thanks again for attending, and good afternoon, good morning, good evening. Bye for now.
Thanks, Stephen.
Bye-bye.
Bye.